Hello everyone, today let's talk about the application of enzyme in dairy industry.
Lactose intolerance happens when body lack enough lactase enzyme to digest lactose properly, which lead to discomfort like bloating and cramps. To solve this, the food industry use beta-galactosidase, or also known as lactase, to hydrolyze lactose into simple sugars such as glucose and galactose. This gives us lactose-free milk which retains all its essential nutrients like calcium and protein. A great world example of this technology here in Malaysia is FONG Fresh Lactose-Free Milk. If you look at the product packaging, it states 0.0 grams of lactose per serving. This proves that the lactose hydrolysis was highly successful, which is achieved by optimizing industrial parameters like enzyme concentration, temperatures, reaction time, which is to ensure complete removal without losing its nutritional quality. Looking closer at the role of lactase, it serves as an efficient biocatalyst. Industrial processing specifically aims to achieve more than 99% lactose reduction to make it completely safe for the consumers. An interesting bonus is that this hydrolysis process naturally increases the milk sweetness, so there's no need to add any refined sugars. Process engineers carefully manage the enzyme dosage and temperature to get this perfect balance. So how does this actually work? Looking at the process flowchart, it starts with the raw milk intake, followed by centrifugal standardization and UHT treatment. Right after that, a sterile lactase reaction takes place. Once the enzyme does its job, the milk goes through aseptic packaging and warehouse storage before finally heading out to the market as a safe, finished lactose-free product.
The lactase, which is known also as beta-galactosidase, will hydrolyze the lactose, which is the main sugar in milk, into glucose and galactose. This reaction is essential because the lactose cannot be directly absorbed by the human body. So lactase deficiency will lead to lactose malabsorption, which can cause symptoms such as bloating, abdominal pain, and also diarrhea. Next, we will discuss about the enzyme mechanism. Firstly, the lactose will bind to the enzyme active site, then the beta-glycosidic bond is cleaved and then releasing glucose. Then a temporary enzyme galactose intermediate is formed. The water will hydrolyze the intermediate and then releasing galactose and regenerating the enzyme. The overall reaction is lactose plus H2O. Outcome is glucose plus galactose. So this picture shows about the detail in the enzyme mechanism. They have mainly two steps, which is step 1, enzyme galactosylation, and then step 2, enzyme degalactosylation. Now we are talking about the sources and industrial application of beta-galactosidase. Beta-galactosidase from different organisms can catalyze lactose hydrolysis through the same mechanism, but they will have differ in optimum pH, optimum temperature, and also the thermostability, as well as the catalytic efficiency. So basically, there's two main sources we use in industrial production of beta-galactosidase. One is yeast, one is fungal. For yeast, its optimum activity is at nearly neutral pH, which is suitable for milk and dairy processing, and it is commonly used for lactose-free milk production. For fungal sources, its optimum activity is at acidic pH, so it is mainly used in dietary supplement and other non-dairy applications.
There are three major industrial limitations associated with the use of lactase in lactose-free dairy production. Firstly, the production cost of lactase is relatively high. The enzyme is produced through microbial fermentation and requires several downstream processes, including extraction, purification, and quality control. This process requires specialized equipment, energy, and skilled personnel, which increase manufacturing costs. As a result, lactose-free dairy products are generally more expensive than conventional dairy products. Secondly, lactase is sensitive to environmental conditions such as temperature and pH. Under unfavorable conditions, the enzyme can undergo denaturation, which changes its three-dimensional structure and reduces its catalytic activity. This decreases the efficiency of lactose hydrolysis and may require additional enzyme supplementation, leading to higher production costs. Lastly, lactase has limited stability during storage and transportation. The enzyme requires controlled conditions, particularly low temperature, to maintain its activity. Over time, its activity gradually decreases, resulting in reduced efficiency and shorter shelf life. This storage requirement increase operational costs and may affect the reliability of industrial process.
In the dairy industry, producing lactose-free milk using the enzyme lactase has some challenges. The main problems are high enzyme costs, unstable enzyme activity, and loss of efficiency during production. To solve these issues, one important method is enzyme immobilization. This means attaching the enzyme to a solid support so it can be reused many times. It also makes the enzyme more stable and longer lasting, which helps reduce production costs. Another method is process optimization, where we control factors like temperature, pH, time, and enzyme concentration. When conditions are correct, lactase works more efficiently to break lactose into glucose and galactose. Good storage and handling of enzymes are also very important. Proper storage conditions help maintain enzyme activity and prevent it from becoming inactive too quickly. This ensures the enzyme can last longer and work better during the production. As a result, lactase breakdowns become more consistent, efficient, and suitable for large-scale milk production. Modern biotechnology also helps improve lactase performance. One method is protein engineering, where scientists modify the enzyme structure to make it more stable and effective. Techniques like directed evolution help create better enzyme versions. Another method is recombinant technology, where genetically modified microorganisms are used to produce lactase. This increases enzyme availability, purity, and reduces costs. In conclusion, lactase or B-galactosidase plays an important role in producing lactose-free milk by converting lactose into glucose and also galactose. Industrial applications such as farm fresh lactose-free milk improve accessibility for lactose-intolerant consumers while maintaining the milk nutrient. Challenges including high enzyme costs, enzyme instability, and also incomplete hydrolysis can be improved through enzyme immobilization, process optimization, and also enzyme engineering. So, these lactase technologies support efficient, sustainable, and high-quality lactose-free dairy productions.